Hello, I'm Lola Fashoyin-Aje, and I'm a medical oncologist and the Deputy Director for the Division of Oncology 3 in the Office of Oncologic Diseases at the FDA. My division oversees the review and approval of therapeutics for the treatment of gastrointestinal cancers, sarcomas, melanomas, and other advanced skin cancers. I'd like to thank the Cholangiocarcinoma Foundation for convening this meeting and for the invitation to participate. I will be providing my perspective on the importance of balancing efficiency with inclusivity in drug development to support approval of drugs that work for all patients. I have no financial conflicts to disclose, and the views expressed here are mine and not to be construed as FDA policy. Today, I will focus my remarks on drug development considerations that dually provide greater access to clinical trials and generate evidence for a broader patient population. As you know, there's currently one FDA-approved therapy for the treatment of cholangiocarcinoma, and that is pemigatinib, which is approved on the basis of a single-arm trial, the FIGHT 202 trial, which was conducted in patients with previously treated unresectable, locally advanced, or metastatic cholangiocarcinoma with an FGFR2 fusion or other rearrangement. FDA granted approval to pemigatinib on the basis of a response rate of 36% that was durable. This drug targets fusions that involve the fibroblast growth factor receptor 2 or FGFR2, and these fusions are found nearly exclusively in intrahepatic cholangiocarcinoma and are detected in about 10 to 15% of patients. There are other drugs in development that also target FGFR2 as shown on this list that I obtained from clinicaltrials.gov website. This list isn't comprehensive, but it reflects active development for this target. Other FDA-approved therapeutic options for patients with cholangiocarcinoma are listed on this slide. These are FDA-approved therapies that were approved really agnostic to the anatomic or histologic cancer type. The approvals are for patients who have solid tumor malignancies that harbor NTRK fusions in the case of larotrectinib and entrectinib, or that are microsatellite instability high or tumor mutational burden high cancers. However, as you can see, a very small minority of patients with cholangiocarcinoma would be eligible to receive these therapies. And so I don't need to emphasize to this audience that there's a paucity of approved treatment options for patients with cholangiocarcinoma, really underscoring that this disease has tremendous unmet need. In the previous slide, I showed you that there's a proliferation of FGFR2 targeted therapeutics in development. It's important, particularly as more drugs are developed, to consider how we can expand the pool of patients who have access to these drugs, either through clinical trials or once they are approved. And as you may know, the American Society of Clinical Oncology, or ASCO, over the past 5 years has convened multi-stakeholder groups that included FDA, industry, advocacy groups, and others to really reassess the clinical trial eligibility criteria and develop a framework for broadening them to be more inclusive and to reconsider some of them. This work was really rooted in the recognition that there has been this immutable statistic that only 3 to 5% of patients with cancer participate in clinical trials. This despite the fact that the number of trials has increased over time and that many trials do not achieve their accrual goals. And one of the primary drivers of this figure of 3 to 5% is that over time, eligibility criteria have become more and more restrictive. There's also been a tendency to copy and paste from trial to trial and reuse previously used eligibility criteria, even when they are not based on sound scientific or clinical rationale. And often when doing so is really not relevant to the disease or the drug under study. From a regulatory perspective, the regulations do not specifically provide mandates on what would be acceptable or unacceptable eligibility criteria. However, an approval must be predicated on data that is pertinent to the enrolled patient populations and relevant to the U.S. population and medical practice. Another point is that the assessment of eligibility for a number of FDA expedited programs, which are really aimed to provide greater interaction between sponsors and FDA to expedite and develop drugs efficiently, really depend on the population that's studied to determine whether the drug in question meets the criteria for filling an unmet medical need setting. And from a drug development perspective, there are benefits and risks to broadening eligibility criteria, as I've shown on this slide. On the one hand, broadening eligibility criteria provides early access to investigational agents and may actually shorten timelines for accrual, which is really beneficial to both patients and investigators and sponsors. Broadening can also provide the opportunity to get a more complete safety and efficacy database that really informs clinical practice and can help us identify very early drugs that may not be effective or as effective in subgroups of the population. However, there are also risks, as greater heterogeneity in the study population can really be challenging for the interpretation of study results. It may be more difficult to identify an efficacy signal, particularly when the drug's effects aren't very large. And it may be difficult to interpret the safety, knowing what adverse events are due to the disease versus to the drug, particularly in single-arm trials. There may be safety concerns as well that require more frequent patient monitoring. Overall, however, the aim really should be to achieve balance between the need to maximize opportunities to isolate the drug's effect in a clinical investigation and ensuring that the results of the investigation are informative for the context of use. That for an approval, demonstration of substantial evidence of effectiveness really occurs in the context of the population that will use the drug. However, we've seen that broad adoption of the framework that's been set forth by ASCO and in the FDA guidances that were developed subsequent to the ASCO framework has really been slow to occur. This may be due to the fact that change is difficult, and also that there's a perceived risk in changing things, deviating from how we've typically done things. But also, I think some hesitancy regarding really just how to incorporate this framework into clinical trial conduct. So we at FDA have taken a proactive approach at trying to operationalize the framework provided by the ASCO work in disease-specific context. And we've convened stakeholders who are subject matter experts across specific disease settings, initially in cholangiocarcinoma and in pancreatic cancer. And really the scope of the project is to assess the current eligibility criteria practices in these diseases, identify criteria that are considered important to include in all trials, as well as criteria that are frequently used but which may not be appropriate in most situations or in any situations because they may be more objective ways to characterize a patient's baseline and fitness for the trial that should be used instead. And part of this initiative is also to consider the barriers that are outside the scope of the protocol, but that are clinically relevant. And the aim really is to come up with core set criteria that can be implemented consistently across trials that are serving regulatory purposes. So on this slide, I want to shift gears and I'd like to briefly touch upon the design considerations that may lend themselves to providing broader access to trials and could potentially promote faster adoption of therapies once they're approved. This is by no means a comprehensive list of considerations given the time constraints, but I wanted to start off by talking about the tension between drug development efficiency and studying more diverse populations. I think we recognize that there is the attention there in trying to ensure that we get the trials completed in a timely fashion and get drugs out to patients quickly, but also evaluating as many subgroups as possible in the trials. At FDA, we have encouraged sponsors to consider designing trials that could enroll as a primary cohort upon which the primary analysis could be based, but also enroll a cohort of patients that based upon more relaxed criteria, perhaps even evaluating different dosages, which can really provide valuable information to prescribers and patients once the drugs are approved at the time of approval or shortly thereafter. The second point I want to make is that there should be, particularly in disease settings where there's where the disease is rare, and I think cholangiocarcinoma may fall in this category, that we really encourage the use of platform trials to promote efficiency in trial conduct, but also to use our patient resources in a way that really reflects the investment that they're giving. So there is really, if there's a disease space where there are multiple drugs, some of which are targeting the same target, it may not be appropriate to have several randomized clinical trials, each with a control arm, when we could use a platform trial, for example, and have a common control to which each of the drugs can be compared. So we really are promoting that as a way to conduct these development programs in an efficient manner. And we would interact with any patient, any sponsor with a proposal for this kind of an approach. And the final point I'd like to make is that there really also needs to be a paradigm shift in the way that trials are designed and conducted. The model that we've used in conducting trials in the comprehensive cancer centers or the large cancer centers is not really providing access to patients that is needed. There really needs to be a shift from asking patients to travel miles and miles or sometimes across state lines to find trials, to a model wherein we are actually meeting patients where they are, taking the trials to their backyards, so to speak. There no doubt needs to be a lot of capacity building to really achieve that. And this definitely needs to be addressed. But I think another critical component of that is that we need to begin to think about ways that we can actually design the trial so it is feasible to conduct them outside of these large academic settings. And we began some of these discussions with sponsors prior to the COVID-19 pandemic, but certainly the pandemic has provided opportunities to pressure test these measures. For example, leveraging technology and using telemedicine and e-consent to decrease burden to patients, decreasing the number of trial procedures as appropriate, and really decentralizing some portions of trials. For example, allowing patients to perform their laboratory and imaging tests closer to where they live. As you may well appreciate, during drug development, it's really critical to identify a dose that optimally balances drug efficacy and drug-associated adverse events. In oncology, we've traditionally had a higher tolerance for toxicity and really an addiction to the idea that more drug is better. And we are finding out that this is not always the case, particularly for targeted therapies where you might have target saturation occur across a wide range of dosages. And I think it's really particularly important to consider this, the tolerability of the drug when it's really chronically administered to patients, even in the setting where patients may be experiencing low-grade toxicity. Having that toxicity occur chronically is really detrimental to patients' quality of life. So we have great opportunity to really supplement our traditional ways of assessing exposure response analysis for safety with patient-reported data. And we have been encouraging sponsors to consider using these patient-reported outcome data as they really directly reflect symptomatic adverse events, and to use that as a complementary to the physician-reported adverse events in these analyses to optimize dose. I want to really close by saying that there's really increasing interest at FDA in oncology to really get the dose optimized prior to initiating the trials that would support approval of a drug. And we have been discussing internally potentially putting trials on clinical hold if an adequate dose has not been identified prior to initiating these trials. And so I think it would be preferable that some of these considerations be embraced voluntarily rather than really have to put a trial on clinical hold. So I will conclude by saying that adoption of practices that improve access and facilitate adherence to clinical trials and that help generate data that are generalizable is really critical to improving outcomes for all patients. Sponsors should employ rational and streamlined eligibility criteria, flexible trial design and analyses, and multi-pronged and staged approach to dose optimization. I want to thank the Cholangiocarcinoma for inviting me to talk today, and thank you all for your attention. And I will close by highlighting our Project Facilitate, which is our program that assists providers with requests for access to investigational therapies through expanded access. Thank you.